All right, well, thank you all for coming. This is our second speaker for our ME 520 seminar series, the Mechanical Engineering Department seminar series. Our speaker today is Adam Glaser, who's an acting instructor in the Department of Mechanical Engineering. Adam did his undergraduate at Tufts University on the East Coast, got his undergraduate, his BS in mechanical engineering, and then went to a school called Dartmouth for his PhD and graduated in 2015. And afterwards, he came to UW as a postdoc. He's been working with John Liu from 2015 to 2019. But Adam's a rising star and has been recognized by the National Institutes of Health with a K99 award. This is pathway to independence. It's a funded postdoc and then also funding as a assistant professor when you transition. So it's like a 2-year postdoc, 3-year funding as an assistant professor. But these are really competitive awards. It's quite remarkable and quite excellent that he's been able to get that. But that's not the only thing he's been up to. He's also been quite productive in his own efforts with startups. So he's a co-founder in Dose Optics as well as Lightspeed Bio. And today he's going to talk about some of the technology that he's developed and the applications it has to biology and medicine. So I'm going to ask that everyone keep your microphones muted during Adam's presentation, but feel free if you'd like to write in the chat with questions you may have. After Adam's done with his presentation, then I will kind of moderate the Q&A session. So without further ado, I'll let Adam take over, and we look forward to hearing your talk today, Adam.
Great, thank you. Thanks for the kind introduction and also just for the opportunity to speak here. And thanks to everyone who's attending. I know that this is kind of strange times and we have to do things through Zoom now, but hope everyone is staying safe and healthy at home. So as Nate mentioned, today I'm going to be talking about the work I've been doing over the last 5 years or so at the University of Washington. And it's mainly been on developing different technological tools, specifically using lightsheet microscopy, as I'll go into in the talk, for clinical applications in the medical setting. Okay. I just want to start with a conflict of interest statement. As Nate mentioned, I have been involved in co-founding 2 different startup companies, and the second one, Lightspeed Bio, is commercializing some of the technologies that I'll be talking about today. And I am a shareholder in Lightspeed Bio. Okay, so the goal of everything I'm going to talk about today is to explore clinical applications of lightsheet microscopy. And specifically, I'll be talking about 3 different topics, which I'll devote, roughly the same amount of time to over the next 45-50 minutes. So the first one is what I'm going to call rapid 2D pathology. Specifically, the clinical application would be intraoperative guidance. So here, what we'd like to try and do is overcome the speed limitations of current pathology. The second topic would be to provide comprehensive 3D pathology. Basically, can we use some sort of new technology to push past the 2D nature of current histopathology, and using this increased amount of information, could we improve patient prognostication? And I'll go into that more in depth later in the talk. And finally, some of the newer stuff that we've been working on is thinking about whether or not we can use the tools we've been developing and push them even further to not just improve pathology, which is kind of the first 2 topics, but actually laboratory medicine, which is more molecular diagnostics of tissue. And so can we develop tools to, for example, improve treatment decision support? And so that'll be the final topic. Okay. So as I kind of mentioned on the last slide, the main motivation is that current histopathology methods are actually limited by century-old technology. And so I'm not sure how familiar everyone on this Zoom meeting is with histopathology, but I wanted to give everyone kind of a primer on what the current workflow is. So the first step is that a patient would go to the hospital and they might have a piece of tissue removed from their body through surgery or through a biopsy procedure, depending on what brought them to the hospital. So once that tissue is removed, the first step is that it has to be fixed using formalin, which is analogous to formaldehyde to preserve the tissue. And then the tissue has to be prepared in several ways for the current workflow. The first would be to get all the water out and dehydrate the tissue using alcohols. And the reason it has to be dehydrated is that you'd actually take that piece of tissue and you'd embed it in paraffin wax, which sounds pretty archaic, but that's the way things are done currently. And the reason it needs to be embedded in paraffin wax is that we need to cut thin pieces off of that block so that we can mount it on a glass slide. And it has to be thin enough so that you could actually see through the tissue. So typically these tissue sections are only about 3 to 5 microns thick. And then once they're mounted on the glass slide, depending on what the clinicians would like to look at, the tissue has to be stained using chromogenic stains. The most common would be this kind of pink and purple appearance that you may have seen, and that uses hematoxylin and eosin, or H&E stains. Now, once the glass slide is produced and stained, then it's actually manually analyzed on a lab microscope, kind of giving this pink and purple appearance down there. So there's definitely some disadvantages to this workflow. The first is that it's very slow. This whole process takes hours to even a day per specimen. It's slide-based. Everything is done on the glass slides, which inherently limits things to 2 dimensions. It's destructive in that you're physically removing tissue from that block and putting it on the slide. And it can be difficult to reuse that tissue in downstream procedures. And it's also non-digital. It's actually just usually manually looked at through an analog microscope. Although in recent years, these whole slide scanners, which essentially can scan the slides, have been FDA approved, but it adds another step to the process. And I'd say adoption of those remains to be seen. Okay, so as I mentioned, I'll just dive right into the first application, which would be, can we perform rapid intraoperative 2D imaging? And so what do I mean by that with intraoperative guidance? During surgery, for example, if a patient had a tumor, the surgeon would remove a mass of the tumor from the patient. And they'd hope that they got everything, but grossly just looking at the tissue, the surgeon really has no idea whether they got everything out of the body. And so what they'd like to do is to be able to assess what's known as the margin of that piece of tissue and figure out, is the margin positive or negative? Meaning, does the tumor extend to the surface of the tissue, which would indicate that there probably is some cancer left in the body and they should go back and get more. But currently, they would like to send the tissue to histology to have that whole workflow I just mentioned performed so that the pathologist could then go back and tell them yes or no in terms of re-excising more tissue. But there's a challenge. As I mentioned, the histology workflow currently can take hours to even a day, and that's just simply too slow. You can't leave the patient on the surgery bed for that long, and so this typically isn't done. So what we'd like to imagine is, can we address this clinical need with a rapid 2D pathology workflow? And what would we envision? We'd like a simple preparation where we just take the tissue directly from the surgeon and we stain it with fast-acting dyes just on the surface. And then we do 2D imaging using some microscopic imaging method. No surprise, I'm going to talk about lightsheet microscopy. And then we would like to view the images in real time. We'd have digital data on the computer screen that the surgeon and the pathologist can look at. So our goal would be, can we image the whole tissue surface in, for example, under 10 minutes, as opposed to hours or even a day for the current pathology methods. So, as I mentioned, there's many microscopy methods out there, but what is lightsheet microscopy and what makes it so special? This picture from Wikipedia, I think, does a really good job of simply laying things out. And I think the take-home message is that in most microscopy methods, you have a single objective which you use to deliver light to the specimen and then image backlight. Now, there's some downsides to that. And so the really clever idea of lightsheet microscopy is that you now have 2 paths. One path, which I'm showing here in blue, sends a light sheet through the specimen. And if you orient that at 90 degrees to your imaging objective, you can kind of co-localize this sheet of light right where this thing is looking, and then kind of image it onto a high-speed camera. Now, this is kind of analogous to quote unquote optically sectioning the tissue with a sheet of light as opposed to physically cutting it. Now it seems simple, but it's actually really powerful because by only illuminating a single plane in the specimen, you have very efficient imaging because you're really only looking at the optical section of interest. The other advantage is that this is extremely high speed. So, we're sending the sheet in and we're actually capturing a 2D image of that sheet, which is millions of pixels all at once. And so the speed of light sheet microscopy is really only limited by how fast your camera can go. And so this is really ideal for 3D imaging. And so it's been really revolutionary for developmental biology because it's so gentle to the specimen and it's also so fast. And so I think these are two of the most stunning data sets that I've seen from developmental biology. On the top is a Drosophila embryo, and on the bottom is a zebrafish embryo. And you can actually study the development of these organisms over hours or even days, which I think is quite stunning. And these are just 2D videos, but they're actually 3D data sets. So the amount of data that you can produce and analyze is astounding. So there was a problem for us when we started this project. It's that traditional light sheet microscopes are not well suited for clinical applications. So here's 3 different architectures or geometries for how these microscopes have been built. And the details aren't super important, but I think the take-home message is that they were originally developed for developmental biology, for looking at a single embryo, which is very small, over really long time periods. And so these systems are very good at doing that. But if you imagine trying to take a large surgical resection and putting it into one of these microscopes, it would be very difficult because they currently use cuvettes, which you wouldn't fit the specimen into, or in this case, there's these 2 bulky objectives which come in, and the objectives physically wouldn't allow you to put the tissue here. So the take-home message is that the challenge we realized is that these architectures that existed would not easily be adapted for clinical pathology. And so back in 2017, we produced our first generation, what we call Opentop light sheet microscope. And I think the easiest way to think about what we wanted to produce is just a flatbed scanner for tissues. So here I'm showing that inverted SPIM architecture that was on the previous slide. And all we wanted to do is take this thing and we just wanted to flip it upside down because that would allow us to create, that idea of a flatbed scanner. And so now if you had the objectives below the tissue, you could come in and you could put, a large surgical reception on top, and also, some sort of glass plate or something to hold the tissue. And the entire top would be unobstructed. So this, from an ergonomic and usability standpoint is great. We solved our problem. The challenge is that optically, sending this purple beam and then collecting this green beam, as it's shown here, would just not work because going from air into glass, I think we all know the beams would refract really sharply and we wouldn't get an in-focus image. And so the innovation that we had was to kind of come up with this hemispherical glass element and insert it right here. And if we zoom in more, we call this thing a solid immersion lens. And what it does is actually this curved surface right here is always perpendicular to any ray along the green and the purple cones. And that essentially negates any refraction. So now you get the beams from air into glass without any refraction. And then if you do some clever index matching the rest of the way, you can get in and out of the tissue. And so now we kind of have designed this flatbed scanner, which was really exciting. So here's a CAD diagram of the actual mechanical layout of our first system. On the left here is the optics for generating the sheet, which kind of bends up at 45 degrees right here. And then the opposite arm at 90 degrees images it onto this high-speed camera that I mentioned earlier. On top of it, we put this microscope stage, which allows us to kind of scan the specimen around. And inside that stage, we can take a modular sample holder and put any piece of tissue on top. It's like a scanner. Now, kind of a nuanced point of why our method using light sheet microscopy would be better than, say, a conventional microscope is that these fresh surgical resections in the surgery suite, they have a very irregular tissue surface. There might be little ripples on the surface on the order of tens to even hundreds of microns. It's really hard to flatten it beyond that. And a conventional microscope going just straight up and down has a really short depth of focus. And so that means you're going to miss a lot of the tissue surface inevitably, just due to the fact that it has a really irregular shape. But with our light sheet design, where we send in this sheet obliquely and then we image it obliquely, we actually have a much longer depth of focus, probably maybe 2 orders of magnitude longer. And so we can actually capture this in one shot. And the way we do that is we image that oblique sheet. And that's one of these little black boxes I'm showing here. And then as we move the stage, we just run the camera as fast as we can, and we move the specimen, and we start to kind of build up this parallelogram of 3D imaging data. And so we scan all the way this way until we cover the whole length of the specimen, and then we can move over and we can do the same thing again until we capture the whole surface of the specimen. So this is a video actually showing the system in action. Up here, that is an actual surgical resection, and this is the microscope stage. And if I hit play, let's see, if I hit play, there we go. Here you can see it starting to do that kind of serpentine scanning I was mentioning. In the bottom right, you can see a bottom view of the system. You can actually see that hemisphere of glass that lets us get the beams in and out. And in the very bottom right, you see a raw feed from the camera. It's actually running at 800 frames per second, but that is like that thin volume that captures the irregular surface of the tissue. And you can see that we covered the whole, in that case, the whole surface in about 7 minutes. So if we now look at the imaging data, this is imaging of a human prostate tissue. On the top here, I'm actually showing what happens if you just looked at one 2D plane before we did this so-called surface extraction of that irregular tissue surface. And you'd see that there's these black areas. And those are the areas which would be missed on a conventional microscope. But using our 3D data and performing some image processing, we can actually capture that entire irregular surface and flatten it into a 2D image so that you don't miss any spots. And now if we zoom in, because these are very large gigapixel imaging data sets, you can zoom in on individual glands in this prostate and zoom in further again and see individual cell nuclei. And on the right, we're comparing it to the traditional histology and showing that we can see similar structures. Similarly, this is prostate adenocarcinoma, or these are cancerous glands. And again, if we zoom in on our data sets in black and white, you can see the individual nuclei, and you can compare that to the traditional H&E. And so we did a small feasibility study imaging 25 specimens from the UW Medical Center and comparing if pathologists looked at our black and white data versus what they're used to looking at, how often do they get the same diagnosis? And it was encouraging that we saw there was 92% sensitivity and specificity, indicating that our method actually could be useful in an intraoperative setting for pathologists to interpret in real time. Now, we don't just look at, we don't have to just look at prostate tissue. We can also look at breast tissue. So this is a video kind of zooming in on a data set of a breast lumpectomy specimen. And so we're going to kind of zoom out here. And then you might notice in this, I'll pause the video, in this top right region, we're about to zoom in. In this particular data set, this is actually breast cancer. And so kind of going back to that margin part that I mentioned at the beginning of this particular clinical application, this would actually indicate a positive margin in that the tumor does extend to the edge of the tissue, and that could indicate that there still is tumor within the patient. And so this is just kind of an anecdotal example of what I was discussing before, where this could be used to guide surgeries. Okay, so a little bit of an abrupt change, but I'm going to kind of switch gears now from that first application to the second one, which is something that I've actually spent the last several years working on. So it's a little bit more recent work. And that's on the comprehensive 3D pathology. So kind of comparing and contrasting it back to the downsides of the current workflow, what is a 3D pathology workflow that we would envision? It would start much the same in that there would be collection of some sort of tissue, either following surgery or, for example, biopsies. Those tissues would still be fixed to preserve them, but then the preparation would be different. We wouldn't dehydrate it and we wouldn't embed it in wax. What we'd envision is we do whole mount staining, meaning we'd want to get fluorescent dyes diffused throughout the entire 3D structure of the tissue. And then we do something called optical clearing, which I'll go into more detail about. And what we could do there is we could actually render the tissue completely transparent, like a piece of glass. And that really is what allows us to do 3D imaging. And then finally, we do 3D imaging. You could use any microscope, although it would only really be feasible with lightsheet microscopy due to the speed that I mentioned before. And so we'd envision doing 3D lightsheet imaging. And finally, digital analysis. These data sets, extending them from 2D to 3D, they become very large. And so you'd have some sort of high performance computer to store and analyze the data, and also do volumetric visualization of the data. Now here, the clinical goal is, could we do high throughput 3D imaging of clinical biopsies? And for this application, over the next several slides, I'll kind of dig more into this. Okay, but first, what is optical clearing? I think the easiest way to think about it is optical clearing is really just refractive index matching. So I found this video recently on the web. So what you'll see here is this is just a jar full of like little beads. And what happens is this person pours water into the jar. And what you can see is it looks like it's like an illusion. The beads disappear. And then I don't know why, but you can see this particular person, I guess, put like little Pokemon toys inside the jar. But now you can see those and you couldn't see them before. So I think that's just like, this is just a really simple anecdotal way to kind of understand what optical clearing is. And so, if we draw a little cartoon, you have these plastic beads and they're surrounded by air. But then if you replace the air with water and the water has the same index as the beads, they appear to just disappear. So you can really do the same thing with biological tissues. So here, you have some cells that I'm showing in tan, and they're surrounded by water because our bodies are about 2/3 water. And so the cells in the water don't have the same refractive index, but you can play the same trick. So you can actually remove the water and then you can put in some solvent, some chemical that has the same index as the cells, and then you get the same effect. And so this is actually an example from the lab. This is a large piece of human tissue, about several centimeters on each side. And then after clearing, you can see that it is rendered transparent and you can actually see the grid through it. And so, I think this is just a basic, it's really just a basic principle of how this optical clearing works. So, going back to pathology, why would this be exciting? I think there's several clinical questions which could really be helped and aided by 3D pathology. So these are just three kind of toy examples that or categories that I broke things into. But one would be so-called convoluted structures or complex distributions of cells. And the final one would be sparse or rare objects or cells. So starting with convoluted structures, these are all 2D views. These are examples of what you'd see on a glass slide. Now, if you look at the convoluted structure one, it's hard over Zoom. I would normally ask, like, can anyone tell me what this would look like in 3D? But it's really hard. And if you actually looked in 3D, you'd see that it could be this, complex intertwined network of branching tubes, but it's really hard to tell that from the 2D view. Similarly with the complex distributions, from this 2D view, you'd think that the blue cell is closer to the red cells and the green cells are far away. But if you look in 3D, you'd actually notice there's a lot of green cells right around here. They're just not being captured on that one 2D view. And finally, for sparse or rare objects, you might see nothing in 2D, but actually in 3D, there could be somewhere sparsely distributed in that piece of tissue, just a few of these rare cells. And so there's actual biological examples and basis for these. Convoluted structures could be vessels, lymphatics, or glands. Complex distributions could be, for example, immune cells and understanding the spatial relationships between immune cells. And finally, sparse or rare objects could be these rare metastatic cancer cells, which are often the drivers of death in cancer patients because they drive metastasis, but they're just very difficult, if not impossible, to find on current 2D glass slides. So some of the first 3D imaging work that we did was of human prostate biopsies, and this was with Nick Reeder and Larry True in pathology. So this is just a cell phone picture of the biopsy before and after clearing, and you can see how now it looks quite transparent. And if you sent it to histopathology, you might get this one glass slide of that biopsy, and you could zoom in and do the diagnosis. But with our light sheet microscopy methods and clearing, you can actually get a rich 3D data set. Here I'm showing a volume rendering, and then these are the 3 orthogonal cut planes through that data set. And so what's interesting is if you now look at one region of interest in our data, you can actually play a video where you're scrolling through the depth of the tissue, and you can appreciate how the glands and the biological constituents of the tissue are changing with depth. And so what we did is, in this anecdotal study, we had Nick and Larry look at this video. We scrambled the depths so that it didn't look like a continuous video, and we had them grade the cancer, give a grading for the prostate cancer. And that's what this graph down here is. You can see that as a function of depth, what's interesting is that the grade, which is known as the Gleason pattern, for each pathologist, they changed their answer depending on what depth they were looking at. And they also didn't agree with one another. So I think both of these findings are interesting. And clinically, this is important because a Gleason pattern 3+3 clinically means that the patient would go under active surveillance, meaning that they would be sent home and just continue to be monitored to see if the disease progresses. Whereas if the grade was a 3+4, they would be sent for a radical prostatectomy, meaning that they would have surgery and their entire prostate would be removed. And these are obviously very different clinical outcomes. And so it's kind of frightening how much variance there is both between pathologists and as a function of depth. But what's really encouraging is when we showed them both the video continuously, like we're seeing here, they both agreed that it should be a Gleason pattern 3+3, meaning that this patient should not have surgery and they should be sent home. So this was really encouraging initial results, but the feedback from both of the pathologists was that ideally we need to image smaller and also deeper so that they can be more confident in their diagnosis. And we as engineers also wanted our system to be, or our next system to be, compatible with more clearing protocols. And so a lot of this work was enabled by John Daniels at Applied Scientific Instrumentation in a microscope objective that he designed. And so the first thing we wanted to do was to image smaller. And so to do that, we wanted to improve the lateral and axial resolution of our microscope. So here I'm showing where our first system was. So we had about 7 micron axial resolution and about 2 micron lateral resolution. And based on the feedback from the pathologists, they said, ideally, we'd be able to achieve about a 20x objective resolution. That's what they're used to looking at. And in terms of axial resolution, we wanted to get between 3 and 5 microns because that's how thick a tissue section that's put on a glass slide is. And so with this new objective, we're actually able to achieve both of those things and fall right in this target zone. We also wanted to increase the imaging depth. On our first system, we had that hemisphere of glass, so we actually couldn't lower specimens to image deeper into them. And that system was actually never designed for 3D imaging. It was really meant for the intraoperative case. But our new system, with this in mind, we extended the imaging depth to 5 millimeters. And finally, in terms of refractive index compatibility, the first system was locked into whatever refractive index that hemisphere of glass had, which happened to be 1.46. But our new second system is compatible with aqueous samples at 1.33, all the way up to 1.56, which is about as high as these clearing protocols go. And so here I'm showing the design of our new multi-immersion Opentop system. It's very similar to the first system, but now we've replaced the hemisphere of glass with just a large liquid reservoir. And then we have this custom objective that seals into the chamber. And so what that allows us to do, if you look over at this kind of cartoon on the right, is now to do our 3D scanning. We can scan the tissue laterally, but we can also lower it to image deeper and deeper into the tissue. This new system, not only achieved the higher resolution that we were after, but also the deeper imaging. So this is again a real life video of the system in action. And I think another important feature of our Opentop microscopes is because they're completely unobstructed on the top, you can either place one large piece of tissue, or you can also place many smaller pieces of tissue. And in this case, I'm showing 12 prostate biopsies. And so if I play the video, you can see the scanning begins. And now we're kind of scanning along the length of one of the biopsies. And if you look in the bottom left here, that red box is showing the depth that we're imaging, and then it'll speed up. But we can set up a high throughput imaging scenario where there's no user intervention other than setting things up, and then we just rapidly, over hours, can image all of these things in 3D. And so, many hours later after imaging and processing, we end up with the data that I'm showing on the left here. So these are the 12 biopsies that were in that chip. And these are actually, terabytes of data and also 3D data sets. So if we zoom in on, for example, biopsy number 8, and now we look at the 3 cut planes, you can appreciate that this is true 3D data. And now if we zoom in on one of the glands, you can see that in all 3 directions, we do have true 3D data. And you can even start to see subnuclear features, indicating that we did achieve the higher resolution that we were after. And then this is kind of just a fun volumetric visualization of one of the biopsies, but you can kind of zoom in as if you're on like a spaceship or something, and you can zoom in on this biopsy and really start to appreciate how complex the information that we can get is. So I'll pause it because it's probably a little choppy on Zoom, but if I pause it here, you can start to see all of the individual cells, and you can start to get a feel for the true 3D structure of this data set and the true 3D pathology that's within this biopsy. And so, again, for the pathologist to interpret the data, we don't want to look at it in kind of the black with neon green and neon blue, but we want to convert it to what they're used to looking at, which is this pink and purple appearance. And so we can take a subset of the data, and we can generate now, again, a video that scrolls through the depth of the biopsy. And I'm not sure what the resolution is like on your screens through Zoom, but I hope you can tell that it is higher resolution imaging data than we had from our first microscope.
It looks great from my end.
Okay, great. Yeah, the videos is always an unknown, I guess, through the Zoom, but I'm glad it is high quality. Okay, so if I back up here, this Z-Stack video, it looks great, but this was something that we processed either in MATLAB or Python, and it takes a while to make these videos, and then it's not practical for the clinicians to have to look at the data in this way. So what we really need is an interactive tool for them to be able to view the data in. And so this is work actually through the startup company, also in collaboration with DR Vision and Luciano Lucas. They're over in Bellevue. And so what we wanted was an efficient on-the-fly kind of H&E rendering software with not a lot of complicated buttons that a clinician could use to actually interpret this data. And so this is kind of preliminary results, but this is just a user. This is an example screen capture of a user in the software, and you can switch the cut plane. So now they're looking kind of bird's eye view downward, which is like the video on the last slide. And so you could imagine this is how a clinician might interact with this really complicated 3D data. And it remains to be seen how useful, true 3D volumetric visualization is, but there is the ability to also perform that analysis. And so I don't think clinicians would initially be used to viewing the volumetric data, but it'll be interesting to explore how much value there could be there. And so, kind of developing everything from the microscope to acquire the data to, and the optical clearing methods to enable it, and then finally the software, all of these have been built to be high throughput so that we can enable a well-powered clinical study. And so what I mean by that is in John's lab, we'll soon be undertaking a study where we'll take 6 prostate biopsies per patient, 200 total patients, which is going to be 1,200 prostate biopsies. It takes about an hour per biopsy. That's 1,200 hours of imaging, and it's about a terabyte per biopsy. So that's 1.2 petabytes of raw data. So if these methods had not been built high throughput, we would never be able to do this scale of a study. And so I think the tool development that we've done is actually now enabling this. And the overall goal of this study is to really quantify and prove that 3D pathology has value over 2D pathology. I mean, it seems obvious that more data would be better, but we need to quantitatively prove that. And so John and Larry have recently gotten a grant to do this large high-powered study. And so it'll be exciting for us to all see the results in a few years and how much value there is. Now, I just want to quickly mention that, everything I've been talking about so far is cancer related, but we did use this microscope for other things. For example, neuroscience in collaboration with the Allen Institute. We can look at mouse brain tissues. So here we can put slices onto our system and we can image the entire mouse brain slice and see the individual neurons. There's also applications in immunology. So this was work with Michael Gerner over in UW Immunology, looking at lymph nodes, so we can map the locations of different cells. We can also image whole mouse hearts and also whole mouse lungs. And finally, this is just kind of, I think, pretty eye candy, but with UW chemistry professor Josh Bond, he's pioneered a development called expansion microscopy, where they can physically enlarge tissues to increase the resolution of a microscope. So rather than pushing resolution, just blow up the specimen and make it bigger. And so this was a data set of a slice of mouse kidney. And on the right, you can see a volume rendering where we're cutting through the tissue. And we have increased resolution due to the expansion technique. And you can see the individual tubules. And here's a large vessel. And also these green things are glomeruli. Those are the filtration units of the kidney. And so, there are non-cancer applications of this technology. Okay, now moving just into the final section of the talk, I'll talk about the third application, which is seeing whether we can use light sheet microscopy to improve molecular diagnostics. And so just to bring back the conventional histopathology workflow, I won't describe it again, but the whole process essentially produces several glass slides. That's kind of the output of current histopathology. But clinically, that's not where the story ends. So once these slides are produced, it might be that the clinicians have unanswered questions, or they would like to test the molecular aspects of the tissue to guide treatment. And so what happens is the next stage is that these specimens are sent to laboratory medicine for molecular testing. And so, several of the glass slides would be sent to that department. There would be some sort of extraction from those glass slides, maybe DNA, RNA, or protein, and then finally some sort of analysis. So if you look at the workflow in this kind of trajectory, there's disadvantages for laboratory medicine in that it has to be slide-based because that's what histopathology produces. And I mentioned earlier that the whole process is physically destructive to the tissue, but it's actually also destructive to the molecular constituents of the tissue. So particularly the fixation, the dehydration, and the embedding in wax can actually, to a large degree, destroy both DNA and RNA. And that makes things for laboratory medicine quite challenging. So to just kind of quickly summarize some current extraction methods that are used, when the whole tissue specimen is removed, it gets grossly cut into like millimeter thick pieces. And there's a method called cell sorting that people may be familiar with, where the entire piece of tissue is actually digested and all the cells are dissociated. And then if this is important, if you know what you're looking for, you can fluorescently label those cells now that they're all just floating in some sort of media. And then you send them through a cell sorter that looks for the fluorescence that you labeled in the cells of interest. And then it either rejects cells that aren't fluorescent, or it keeps the fluorescent ones so that now you have a purified volume of only what you want. And then you can do downstream analysis. Although this is really undesirable because one, you destroy all the tissue, and two, you already have to have a label for what you're looking for. So two other methods are based on the glass slides that I mentioned. So now, the tissue could be sectioned and sent through histology. And there's a technique called microaspiration where you can actually come in with a pipette and suck out one individual cell from the monolayer on top of the glass slide. Similarly, there's something called laser capture micro-dissection where you actually use laser light itself to pull the cell out. And in both cases, you have now these individual cells. You can identify them structurally, so you don't have to know the molecular characteristics. And then you can send them for downstream analysis. So what would we, what are we envisioning? We're going to call it light sheet micro-dissection for now, but what we'd like to do is jump into the workflow here, take the whole piece of tissue, but do a non-destructive preparation. We're not going to dissociate it. We're going to clear it and label it like we've been doing before. We'd like to do, these are going to be so big that we'd have to do low magnification screening to find interesting regions. Otherwise, the imaging would take too long. But we'd like the microscope to then be able to go back at higher magnification to confirm the regions of interest. And then finally, some sort of way to extract the cells and send them for analysis. Now, where would this be useful? If I go back to those three anecdotal categories of 3D pathology being useful, this would really be useful, you can imagine, for sparse or rare cells where you don't find them on 2D sections. And you could only find them if you actually looked at the whole intact 3D specimen, specifically rare metastatic cells. And so, looking back to the engineering side of things, there's a technical need. For now, we need a multi-resolution microscope capable of imaging from the meso scale to the submicron scale. So here I'm plotting on the x-axis, what was the lateral resolution of the microscope? You can see the first and second system. And then on the y-axis, it's just a trade-off. What was the imaging speed? And so you can see as you go higher in resolution, the imaging becomes slower, which makes sense. And so what we really wanted was, as I mentioned, fast mesoscopic screening, at say 1 centimeter cube per minute, very fast. And this is where our new system falls. And then we also want slower but high resolution imaging so that we can confirm the interesting regions that we identify at lower mag. And so our new system kind of spans, or can span, almost this entire continuum, which is really important. And I won't go into the details, but this is just a diagram showing how the next microscope will work. Again, we send in a light sheet at an oblique angle, but we actually image it not at 90 degrees, but we image it down into a high resolution path. And that allows us to stick another second lower resolution path on the other side. So now we have this kind of hybrid microscope. And we can, lock the entire thing into a chamber just like we did in past systems. So these are just some very preliminary results, but here we took a larger piece of tissue and we preserved a modified fixation that better preserves RNA and DNA. We fluorescently stained and cleared the tissue and got it to this point. And then we mounted it on our microscope and we did the imaging. And so using the multi-resolution kind of imaging protocol, we could screen this entire large, centimeter-sized piece of tissue at 5x, identify interesting regions, go in at 10x or even 20x. And in this case, we were able to identify cancer cells that have invaded the vascular space, which is known as lymphovascular invasion, or these are actually metastatic cells, kind of as I was talking about before. Now, the second part is that we want to take them out. So once we find them, we want to take them out for molecular analysis. And so what we'd like to do is our Opentop microscopes, again, we have an unobstructed top region so we can kind of interface accessory technologies into the system. So here's a cell extraction system from a collaborator. I'll show more details on the next slide, but basically we'd like to connect that to the top of our microscope. So this is, I think, a really impressive video. This is from Tarang Gujral, a collaborator at the Fred Hutch. This is a robotic dual pipetting system that I showed on the last slide. Now this is just attached to a commercial microscope, but again, we'd like to put this on our light sheet system. But what happens is it's completely automated, but the two pipettes, well, you'll see in a second, they slide in and then they both inject into the tissue. And what's happening at the microscopic scale is on the left here, or actually on the right, is this delivery pipette. And what it's doing is it's delivering a lysis buffer, which will pop the cell. It's delivering that into the cell of interest. And then the other side, you'll see after the cell pops, is the extraction side, and it sucks out the contents of the tissue. And so we're hoping to kind of combine and merge our technology with this technology to provide the cell extraction side of things. And finally, just looking to future directions, I think another kind of untapped area that we'd like to go into is 3D mapping of many cells with moderate molecular resolution. And again, going back to the 3 examples, I think this falls into the complex distributions of many cells sort of category, where you have, it's not feasible to suck out all of these cells, say hundreds or thousands. With what I showed on the last slide, it would simply take too long. But what if you wanted to do molecular mapping of these cells and get a reasonable number of molecular targets? So what we'd envision is, again, we can put accessory technologies on our microscope. In this case, we could put some sort of microfluidic device with syringe pumps. And what we'd like to do is we'd like to pump different labeling reagents into our tissue in an automated and controllable way. And we would just repeat this process several times. And what that would allow us to do is, if we look at the graph on the right, on the x-axis, I'm showing how many cells can we look at. And on the y-axis, how many labels or how much molecular resolution do we have. So the cell extraction technology I mentioned in the previous couple of slides can really only be used for maybe 1 to 10 cells, but it has really high molecular resolution because you can send those cells for example, sequencing. But there's this kind of untapped region where there's lots of cells, and what if you only wanted moderate molecular resolution? And I think that's where this technology would fall, where using the fluidic device, we could label the tissue, we could strip off the dyes, we could relabel the tissue again to look at new things, strip the dyes, label again, and you could repeat this in a certain fashion where, how many times you do it will just keep raising up this circle to more and more molecular resolution. But realistically, you might fall somewhere here where you could get 10 to 100 labels over thousands of cells. And so I think this is an exciting future direction that we could take our technologies into. And finally, just to conclude, everything in this talk, the title was clinical, but it should have really been cancer, because everything we talked about was cancer related. But these tools are really applicable outside of cancer research, which I think is also an untapped area. And so we do have, preliminary data and collaborations with several researchers within UW, for example, immunology with Michael Gerner that I mentioned before. Also with neuropathology, looking at Alzheimer's research, imaging the brain tissue of Alzheimer's patients in 3D to better understand what initiates or causes Alzheimer's disease. Also ophthalmology, studying macular degeneration. This is just preliminary data we obtained imaging whole mouse eyes. Again, at the Allen Institute, looking at mouse brains to do sort of 3D connectome mapping. And then finally, just a recent collaboration starting with PBio, Oscar Vivas, and I mixed their names up. They should be switched, but this is Oscar and this is Claudia. And they're interested in studying the pacemaker of the heart to understand some of the basis of aging within the heart. And so these are, kind of ripe areas where we could take these technologies out of the specifically cancer-focused research. So with that, I'd just like to conclude and acknowledge, all of the people that have helped with this work and also mentored me, specifically John Liu from here in UW Mechanical Engineering, who's really mentored me over the last 5 years. And it's been great and exciting to work with him. Also, Nick Reeder and Larry True in pathology, who've really been an integral part of a lot of this research, as well as our funding sources. Thanks so much.
Thank you, Adam. I think if people want to, they can give the applause reaction on their screens, or I can maybe just, there we go. There's some applause. That's excellent. Thank you, guys. It's a very quiet, subdued applause, but maybe they're applauding elsewhere. Yeah, no, that was great. That was an excellent talk. I think there's questions. I didn't see any questions coming in the chat, but I guess I can lead off with some questions if people want to go ahead and either use it in the chat feature, or they can also unmute themselves and go ahead and speak. But my question is, in your, you showed the system where you guys had both the high resolution objective and the low resolution objective kind of being tightly positioned within that.
Yes, yeah.
What was the purpose for having both the high and the low in your setup?
Oh yeah, sorry I sped through a lot of that section. So the the goal there would be, I had that graph where in 3D, the imaging times and the data set times scale with the cube of the resolution just because things are in 3D. So to image a whole specimen that's really large at high resolution would be, like, you just couldn't do it because it would take too long. So you'd want to image it at the lower resolution scale. But then at low res, it's hard to be confident that what you're looking at is truly what you want. So you do want to go back and zoom in and see individual cell nuclei or even subnuclear features. So you kind of need to have both. And ideally both would be in the same system so that everything stays co-registered. So you can automatically switch from low res to high res.
I see. Question that why do you need two different objectives? Why do you need a high resolution objective and a low resolution objective?
Oh, yeah. Okay. Yeah. Sorry. And the reason that you need two separate ones is there's a trade-off in microscope objective design where as the resolution gets pushed higher, the field of view of the objective just gets smaller and smaller. And that's a pretty standard trade-off. So, a high resolution objective might have a 0.5 millimeter field of view, which is pretty tiny, whereas a low resolution one might have a 5 millimeter field of view. And so kind of optics-wise and technologically, it's not possible to do everything in one objective.
I see. Okay. So Ariana had a question here. If I can either read the question to you, or I can have Ariana unmute themselves and speak if they'd like.
I think I have it up. Yeah, I have it on my screen. So the cost of imaging with our system versus the industry standard. Do you mean like the actual cost of running one experiment or the actual cost of the system?
Probably.
I guess I'm asking, like, if they were to put this in clinical trials right now, or if patients were getting this, how much would it cost the patient to get this test versus the industry standard?
Yeah, so that's a great question. And so we've done an analysis of that, or Nick Reeder, who's the CEO of our startup company, has done extensive analysis of this. So in terms of actual reagents used to do our imaging versus the standard imaging or the standard pathology, it's actually less expensive in that producing glass slides is actually quite expensive, and you need to pay dedicated personnel like a histotech to cut the tissues. That's actually like skilled labor. So it would be cheaper from that standpoint, but beyond that, going into like the reimbursement and the actual medical healthcare, how things would be handled there, I mean, that would remain to be seen, but I don't see any reason why it couldn't be cheaper or at least the same cost.
Cool, thank you.
Yeah. Another question on the accuracy at which the grayscale images are converted. What kind of value? Yeah, so that's a great question as well. So
sorry, the question wasn't on the group chat. It might have been just a private chat. So you mind actually reading the question?
I'll read it. This is from Yashwanth. A quick question on the accuracy at which the grayscale images are converted to conventional H&E stains. What kind of validation was done? So yeah, I didn't go in depth, but again, what we can do is take the grayscale images of fluorescence and then using image processing, we can convert them to that pink and purple appearance. So I'd say that the extent of the clinical validation there was simply having our pathologist collaborators view our processed images and see whether they can interpret them. In terms of the accuracy, we didn't do anything specifically, but if anything, the clinicians were much more accustomed to viewing the converted images. There's definitely a learning curve if you just show them those black and white images. And it's a qualitative result, but the pathologists we've worked with have said that they would pass like the pathologist Turing test, and that many pathologists might not even know that they weren't real images. So they are that realistic, I'd say.
That's cool.
Maybe I'll add that we are just about to submit a paper on this where we tried to quantify the color space of our false colored images. And we found that we can do it with a lot more consistency compared to glass slides. There's actually a lot of variability between labs and even within a single lab at different times. The coloring, if you go on Google and you just search for pathology images, you'll see quite a wide range in terms of coloring, different pinks and different purples, different levels of saturation. So by doing it digitally, actually, we can control it more. And we found that in the paper about to submit.
Yeah, that's a great point.
Could you comment a little bit about the sort of design space? I mean, this is sort of like robotic surgery in some sense, where you have to kind of get all of these pieces jammed into a really small space, you know? So in terms of like, it seems, is there still room to cram more things in there, or is it, you know, you kind of gotten as far as you think you can get with cramming objectives and illumination all into that top of a microscope?
Yeah, no, I mean, that's in designing these light sheet systems, that's definitely that's probably the first design challenge is how do you fit these bulky objectives, and you know, they all have certain working distances, so you can't really you can't change that. So how do you jam them all together? I'd say that dual collection path system I showed at the end, you can see everything fits a little bit tighter now that we have 3 in there. But I'd say there's more possible if custom objectives are made. And so we definitely have a close relationship with this applied scientific imaging company. Both of our systems use custom objectives from them. And so, you know, depending on where the field goes and what the needs are from ourselves and other researchers or clinicians or even companies, more will be possible as more and more custom objectives are made, and that allows researchers like ourselves to continue innovating on the design of these systems. And then finally, on top of the microscope, you know, we really have only started recently diving into what do we do with the whole open top that we have. That's really the big advantage of ours. And so cramming these accessories on, like the aspiration system or microfluidics, I think that's a ripe area for enabling future innovations and future studies.
Yeah, no, I definitely agree that the Opentop is a huge freedom, because you now have a lot more of experiments and robotics that you can then, as you kind of showed us with the robotic pipetting system, and also like the microfluidic designs as well. So that's really nice to be able to have that freedom and not be constrained by, you know, whether it be like the phase light illuminator or other things that are usually kind of getting in the way of trying to do your own stuff on top.
Yeah, exactly.
Are there any other questions from our audience?
Oops.
All right, well, if not, then I want to just thank Adam again for giving a great talk, and thank you guys all for attending. And if people need to reach out to Adam, his email address is also listed there in the lower right corner. But thanks again, and we'll see you guys in a few more weeks for the next talk. Thanks so much.
Thanks so much.
All right, bye everybody.
Bye, guys.